Hello there, and welcome back to Bedtime Science. I'm really glad you're here tonight. Below the surface of the ocean, past the sunlit shallows where coral reefs grow and dolphins feed, past the twilight zone where light fades to near nothing and creatures carry their own illumination, there is a world that covers more than half of the Earth's surface and that remains less mapped in detail than many places beyond our planet. The deep sea begins at approximately 200 meters depth and extends downward to the floor of the ocean trenches, where pressures exceed 1,000 atmospheres and temperatures hover just above freezing in perpetual, total darkness. It is the largest habitat on Earth. It contains ecosystems unlike anything at the surface. It is home to organisms whose biology, chemistry, and behavior break the rules that govern life in the sunlit world above. It has remained largely unexplored not because of indifference, but because of the extraordinary technical difficulty of reaching it. The pressures that would collapse air-filled spaces and make ordinary human survival impossible. The cold that would kill in minutes, and the darkness that renders ordinary observation impossible. What we have learned about the deep sea, accumulated slowly over more than a century of difficult and expensive expeditions, is astonishing. What remains unknown is almost certainly more astonishing still. Tonight we descend slowly into it, not as a horror story, and not as a catalog of monsters, but as a quiet scientific descent into the largest hidden landscape on Earth, a place where darkness is ordinary, pressure is constant, and life has learned to make light for itself. The deep ocean is defined in various ways, but a common scientific boundary begins near the depth where sunlight can no longer drive significant photosynthesis, around 200 meters in clear open water, though the exact depth varies with location, season, and water clarity. Below this depth, the ocean is divided into further zones based on depth and the conditions that prevail there. The mesopelagic zone, from 200 to 1,000 meters, is the twilight zone, a region of extremely dim, diffuse light where faint sunlight still shapes vision and behavior near the upper boundary, but where there is not enough light for ordinary photosynthesis to support an ecosystem. The bathypelagic zone, from 1,000 to 4,000 meters, is the midnight zone, complete darkness, near freezing temperatures and pressures between 100 and 400 atmospheres. The abyssal zone, from 4,000 to 6,000 meters, covers a vast fraction of the seafloor, an immense dark landscape larger than all continents combined. And the hadal zone, below 6,000 meters, exists only in the ocean trenches, narrow channels plunging into the earth where tectonic plates have subducted, reaching their greatest depth in the Challenger Deep at approximately 10,935 meters. Light in the deep sea is not simply absent. It is replaced by a completely different source of illumination: bioluminescence, the production of light by living organisms through chemical reactions. In surveys of midwater animals, roughly 3/4 to 4/5 of observed animals in the mesopelagic zone are bioluminescent, making the deep sea the most extensively light-producing environment on Earth. Bioluminescence is not one single chemistry used by every species, but many systems involve a light-producing molecule, often called a luciferin, and an enzyme or protein system that releases energy as photons of light rather than heat. Different species have independently evolved bioluminescence dozens of times throughout the history of life. It has arisen in bacteria, dinoflagellates, jellyfish, squid, fish, crustaceans, and many other groups, making it one of the most frequently evolved traits in the history of life and suggesting that the production of light in darkness confers powerful survival advantages. In the deep sea, bioluminescence serves as camouflage, communication, predator attraction, prey luring, and alarm signaling, a complete visual language written in light in a world where no other light exists. The pressure in the deep ocean is one of its most defining physical characteristics. Pressure increases by approximately 1 atmosphere for every 10 meters of depth. So at a depth of 1,000 meters, the pressure is approximately 100 atmospheres, equivalent to 100 times the atmospheric pressure at sea level. At the bottom of the Challenger Deep, the pressure is approximately 1,100 times the atmospheric pressure at sea level, roughly 1,100 atmospheres, or approximately 11,000 tons of force per square meter. This is enough pressure to compress water by approximately 5% compared to its surface volume, to crush most materials not specifically engineered to withstand it, and to require the most robust engineering in the most carefully designed submersible vehicles. The organisms that live at these depths have evolved structural adaptations to function under these pressures, including the absence of gas-filled spaces that would be compressed catastrophically, and the presence of biochemical adaptations that maintain the fluidity and function of cell membranes and enzymes under the extreme mechanical load. The temperature of the deep ocean is extraordinarily cold across most of its extent. Below approximately 200 meters depth, ocean temperature drops rapidly through the thermocline, the transition zone between the warmer surface waters and the cold deep, and then remains nearly constant at approximately 1 to 4 degrees Celsius throughout the abyssal and hadal zones. The cold is not uniform everywhere. In the polar seas, cold, dense water sinks at the surface and forms the great water masses of the deep ocean, while in certain volcanic areas and hydrothermal regions, localized heating can produce warmer conditions. The general deep-sea environment is one of extreme cold combined with extreme pressure, darkness, and very limited food supply, conditions that have shaped the evolution of deep-sea organisms toward low metabolic rates, slow growth, long lifespans, and behavioral adaptations for locating and exploiting rare food resources. The seafloor is covered with sediment that has accumulated slowly over millions of years from the rain of particles sinking from the waters above. This marine snow, the continuous gentle fall of organic particles, fecal pellets, dead organisms, and inorganic mineral grains, settles on the ocean floor at rates of approximately 1 to 10 centimeters per 1,000 years in the deep ocean. Despite this slow accumulation, the sediment layer on the abyssal floor can be hundreds to thousands of meters thick in regions where deposition has continued for hundreds of millions of years. Deep-sea sediment is a remarkable archive. It contains the preserved remains of organisms that lived in the surface ocean over millions of years, and by analyzing their chemical composition, isotopic ratios, and species assemblages, paleoceanographers can reconstruct the history of ocean temperature, chemistry, and biology extending back tens of millions of years, one of the most detailed climate archives available to science. The discovery of hydrothermal vents in 1977 was one of the most revolutionary events in the history of ocean science and biology. The vents were discovered by a research team using the submersible Alvin near the Galapagos Rift Spreading Center, approximately 2,500 to 2,600 meters below the surface. The expedition expected to find a relatively sparse seafloor. Instead, they found structures resembling chimneys rising from the seafloor, billowing water heated by proximity to the magma beneath the ocean crust to temperatures of up to 400 degrees Celsius at the vent opening. Water that remains liquid only because the extreme pressure at that depth raises the boiling point. Around these chimneys, they found communities of organisms in extraordinary abundance and variety. Clams up to 30 centimeters across, tube worms up to 2 meters long, crabs, shrimp, fish, octopuses, and bacteria covering every surface in dense mats. None of these organisms depended in any way on sunlight. They derived their energy entirely from the chemical reactions between the vent water and the surrounding cold seawater, primarily through the oxidation of hydrogen sulfide by chemosynthetic bacteria that formed the base of the food web. The discovery proved that rich ecosystems could be built without direct sunlight, fundamentally changing understanding of the requirements for life and opening the possibility that life might exist in environments, including the subsurface oceans of moons in the outer solar system, previously considered impossible for biology. The tube worms found at hydrothermal vents, Riftia pachyptila, grow to lengths of up to 2 meters and form one of the most unusual biological partnerships known. They have no mouth, no digestive system, and no anus. They are anatomically incapable of consuming food in any conventional sense. Instead, much of their body is occupied by a specialized organ called the trophosome, densely packed with chemosynthetic bacteria. These bacteria oxidize hydrogen sulfide from the vent water and fix carbon dioxide into organic compounds, performing the same basic carbon-fixing role that plants perform in sunlight, but using chemical energy instead of light. The tube worm provides the bacteria with a controlled environment, a supply of hydrogen sulfide and oxygen from its specialized blood circulation, and protection. The bacteria provide the tube worm with all its nutritional needs. The tube worm cannot survive without its bacterial partners, and the bacteria in the trophosome have never been cultured outside the host. This extraordinary mutualism, in which one partner is biochemically dependent on the other for all its nutrition, evolved at the vent communities and represents one of the more intimate and complete symbiotic partnerships in the natural world. Black smokers are the most dramatic form of hydrothermal vent, towering chimney structures that can reach heights of 40 to 60 meters, venting superheated water rich in dissolved minerals including sulfides of iron, copper, zinc, and lead. When this superheated, mineral-rich water contacts the cold surrounding seawater, the minerals precipitate out as fine particles, creating the characteristic black cloud that gives black smokers their name. The chimneys are built from these precipitated minerals, growing layer by layer as new mineral deposits accumulate on the outer surface. The temperature of black smoker vent water can reach 400 degrees Celsius, yet certain bacteria and archaea have been found thriving in the water immediately around the vent openings, adapted to these extreme conditions through heat-stable enzymes and other molecular adaptations that allow their biochemistry to function at temperatures that would destroy the proteins and nucleic acids of any ordinary organism. The broader study of heat-loving microbes, including those from extreme environments, has also changed biotechnology. The famous heat-stable DNA polymerase from Thermus aquaticus, Taq polymerase, was originally isolated from a hot spring bacterium rather than a deep-sea vent organism, but it illustrates the same principle. Enzymes adapted to heat can become essential tools for molecular biology, forensics, and medical testing. White smokers, in contrast to black smokers, vent cooler water, typically between 60 and 140 degrees Celsius, that is milky white in color because of the precipitation of lighter minerals, including anhydrite, barium sulfate, and silica. The Lost City hydrothermal field, discovered in 2000 in the mid-Atlantic at approximately 800 meters depth, is an extraordinary example of a white smoker field. The vents there are driven not by volcanic heat, but by serpentinization, the chemical reaction between seawater and mantle rock, peridotite, that produces heat and alkaline, hydrogen-rich fluid. The chimneys at Lost City can reach heights of 60 meters, and the vent fluids have a pH of approximately 11, among the most alkaline natural waters on Earth. The community at Lost City includes abundant archaea and bacteria, but relatively few animals compared to black smoker communities, and the chemistry of the Lost City vents has been proposed as a possible model for the conditions under which life first originated on Earth. Cold seeps are regions of the deep seafloor where methane, hydrogen sulfide, and other chemical-rich fluids seep upward from sediments and support chemosynthetic communities similar in structure to hydrothermal vent communities, but operating at ambient bottom water temperatures rather than at elevated temperatures. Cold seeps are found along continental margins worldwide, wherever organic-rich sediments release methane through microbial decomposition or where hydrocarbons migrate upward from deep geological reservoirs. The communities at cold seeps are characterized by their extraordinary longevity. The tube worms at cold seeps, Lamellobrachia limesci, can live for more than 200 years, making them among the longest-lived animals in the world, and the seep communities themselves can persist for thousands of years. Cold seep communities include dense aggregations of mussels, clams, tube worms, polychaete worms, and associated predators and scavengers, all dependent ultimately on the chemical energy of the seeping fluids rather than on photosynthetically fixed carbon. Methane hydrates are ice-like structures in which methane molecules are trapped within a lattice of water ice, stable under conditions of high pressure and low temperature that characterize the seafloor of continental margins. They exist in vast quantities in deep-sea sediments. Estimates suggest that the total carbon stored in seafloor methane hydrates globally may equal or exceed the total carbon in all other fossil fuel reserves combined. Methane hydrates can be recognized on the seafloor by their white crystalline appearance and are so rich in methane that a lump of methane hydrate will burn if ignited at the surface, giving them the popular name of fire ice. The potential for methane hydrate destabilization in response to warming ocean bottom waters to release large quantities of methane into the ocean and atmosphere is a concern in climate science, though current evidence suggests the process would be slow on human timescales. Certain microorganisms in the sediments around methane hydrates consume the methane through anaerobic oxidation, converting it to carbon dioxide and preventing much of the methane from reaching the ocean water above. The anglerfish, familiar from its distinctive silhouette with the luminescent lure dangling from a modified dorsal fin, is one of the iconic creatures of the deep sea and the most famous example of bioluminescent prey attraction. There are approximately 300 species of anglerfish distributed across the mesopelagic and bathypelagic zones. The lure is present only in females, who use it to attract prey, and males, in the near-total darkness of the deep sea. The bioluminescent organ at the tip of the lure, called the esca, is colonized by bioluminescent bacteria that produce the light through their own metabolic processes, not through any mechanism in the anglerfish itself. Different species of anglerfish have escas of different shapes, sizes, and light emission patterns, apparently adapted to attract different prey species in different depth zones. Some deep-sea anglerfish can open their mouths wide enough and distend their stomachs to such a degree that they can swallow prey larger than themselves, an adaptation to an environment where successful prey encounters are rare and every opportunity must be fully exploited. The sexual parasitism of some deep-sea anglerfish is one of the most extraordinary reproductive strategies in the animal kingdom. In certain families of deep-sea anglerfish, the males are drastically smaller than the females. In some species, the ratio is approximately 1 to 60 in body length, or 1 to half a million in body mass. The tiny males have no lure and are shaped by one overwhelming biological task: finding a female in a vast, dark environment where encounters are rare. In the most extreme species, when a male encounters a female, he bites into her skin and the tissues of the two animals fuse. Over time, the male becomes increasingly dependent on the female, with shared circulation and reduced independent organs, essentially becoming a permanent sperm-producing partner attached to her body. A single female can have multiple males attached simultaneously. This extreme sexual parasitism is thought to have evolved because encounters between males and females are so rare in the vast, dark, deep sea that any opportunity for mating must be seized permanently. The vampire squid, despite its dramatic name, is neither a squid nor an octopus, though it is related to both. It belongs to its own order, Vampirotuthis infernalis, the only species in its order, and represents an evolutionary lineage that diverged from both squid and octopus approximately 300 million years ago. It lives in the oxygen minimum zone of tropical and subtropical oceans, at depths between 600 and 900 meters, in waters so depleted of oxygen that most animals cannot survive there. The vampire squid has enormous eyes relative to its body size. Eyes up to 2.5 centimeters in diameter in an animal with a mantle approximately 13 centimeters long, giving it one of the most striking eye-to-body proportions in the animal world. It is covered with photophores that can be used to produce light and bioluminescent mucus that it can eject as a defensive cloud. Contrary to what its predatory-sounding name implies, the vampire squid is not a predator of living animals, but a detritivore. It feeds primarily on marine snow, capturing organic particles from the water with sticky filaments. In the oxygen-depleted environment it inhabits, its extremely low metabolic rate and its biochemical adaptations for surviving low oxygen concentrations allow it to exploit resources unavailable to most other animals. The barrel-eye fish, Macropina microstoma, has one of the most remarkable visual systems of any animal. Its eyes are large, tubular, and mounted on the top of its head pointing upward, oriented toward the faint downwelling light from above, where potential prey silhouettes would be most visible. What was not known until underwater footage captured the fish in its natural habitat in 2004 was that the two structures thought to be the fish's eyes, the two dome-like structures on either side of the front of its head, above its tiny mouth, are not eyes at all but olfactory organs. The actual eyes are the two bright green structures inside the transparent, fluid-filled shield that covers the top of the fish's head. The entire top of the head is filled with this transparent fluid, allowing the eyes to move freely within it while remaining protected. The fish can rotate its tubular eyes from pointing upward to pointing forward, allowing it to track prey that has passed over its head without losing sight of it. When the fish was brought to the surface in nets, the transparent fluid-filled shield would always collapse, and the unique structure of the eyes was not appreciated until living specimens were filmed by remotely operated vehicles. The ocean floor is not flat. It contains the world's longest mountain range, the Mid-Ocean Ridge System, which extends approximately 65,000 kilometers through all the major ocean basins. The mid-ocean ridges are the sites of seafloor spreading, where new oceanic crust is continuously produced as tectonic plates diverge and magma wells up from the mantle to fill the gap. The ridge system rises to heights of approximately 2 to 3 kilometers above the surrounding abyssal plain, and its central axis is typically marked by a rift valley, a deep trough created by the pulling apart of the plates. The ridge system is not a smooth feature, but is offset by transform faults, fracture zones that offset segments of the ridge perpendicular to its spreading direction, creating the segmented appearance that global bathymetric maps reveal. The rocks produced at the mid-ocean ridges record the history of the Earth's magnetic field in their magnetic polarity, as the iron minerals in the cooling basalt align with the ambient magnetic field at the time of formation. These paleomagnetic stripes, symmetrically arranged on either side of the ridge, were one of the key lines of evidence for seafloor spreading and plate tectonics. Seamounts are underwater mountains that rise from the ocean floor but do not reach the surface. They number in the tens of thousands globally. Some estimates suggest there may be 100,000 or more seamounts taller than 1,000 meters, though most have not been mapped in detail. Seamounts are formed primarily by volcanic activity, as magma from the mantle builds up a pile of basaltic rock on the ocean floor. Over time, As the oceanic crust moves away from the volcanic hotspot that created the seamount is carried to greater depths and the volcanic activity ceases. Some seamounts have flat tops, called guyots, formed by wave erosion when the seamount was at or near sea level, later submerged as the crust subsided. Seamounts act as obstacles to deep ocean currents, forcing water to flow upward around them in a process called upwelling, which brings nutrient-rich deep water toward the surface and creates regions of elevated biological productivity. The communities around seamounts are often highly diverse and may contain species found nowhere else, biological islands of diversity in the otherwise sparsely populated abyssal plain, isolated by deep water from other seamounts and from continental communities for millions of years. Deep-sea fish have evolved a remarkable diversity of adaptations to living in an environment of very limited food, extreme pressure, cold, and darkness. Many deep-sea fish have large, tubular eyes pointed upward to maximize sensitivity to the faint downwelling light from above and to detect the silhouettes of potential prey. Others are nearly blind, having reduced or vestigial eyes in environments where light is completely absent. Many have extremely large mouths relative to body size, capable of engulfing prey as large as or larger than themselves, an adaptation to food scarcity that ensures every encounter with potential prey can be exploited. Some have bioluminescent organs distributed along their bodies in species-specific patterns that may serve as visual identification in the darkness. The fangtooth fish, Anoplogaster cornuta, has proportionally the largest teeth of any fish relative to its body size, teeth so long that when the mouth is closed, the fish must retract them into pockets alongside its brain to prevent them from piercing the roof of its own skull. The dragonfish, of the family Stomiidae, is one of the most effective predators of the mesopelagic zone and has evolved one of the rarest visual tricks among deep-sea vertebrates: the ability in some species to produce and detect red light. Most deep-sea organisms have visual systems sensitive only to blue light, because blue wavelengths penetrate deepest into seawater and because bioluminescence in most organisms is predominantly blue. Red light is absorbed by seawater within a few meters and is effectively invisible to most deep-sea animals. The dragonfish, however, has evolved both a photophore beneath each eye that produces red light and a visual pigment sensitive to red wavelengths, allowing it to illuminate and detect prey using a searchlight invisible to those prey. Certain deep-sea shrimp that the dragonfish preys upon are red-colored, a color that provides camouflage in the deep sea because red light is absent and red objects appear black in the ambient blue bioluminescence, but the dragonfish's red spotlight reveals them clearly. The hadal zone, the ocean below 6,000 meters, existing mainly in trenches, is one of the most extreme and most poorly studied environments on Earth. Despite covering only a small fraction of the total ocean floor area, the trenches receive disproportionate interest from researchers because they include the deepest places on the planet and because the organisms that live there must cope with the most extreme pressures known to support complex life. The Mariana Trench, the deepest of the ocean trenches, plunges to its maximum depth at the Challenger Deep, where the water column above exerts a pressure of roughly 1,100 atmospheres on anything at the bottom. In 2012, filmmaker James Cameron descended to the Challenger Deep in a specially designed submersible, the Deep Sea Challenger, becoming the first person to do so alone and the third person to do so at all. Cameron spent approximately 3 hours at the bottom, observing the terrain through a viewport and using cameras and sampling equipment to document the life and geology of the hadal floor. Life in the hadal zone includes organisms that must maintain cellular function under pressures that would distort the enzymes and membrane structures of surface organisms. One important biochemical adaptation involves piezolites, small molecules including trimethylamine oxide, commonly called TMAO, that accumulate in the tissues of many deep-living animals and help stabilize proteins under pressure. In fishes, TMAO concentrations generally increase with habitat depth, and the related breakdown chemistry contributes to the strong fishy odor that appears after tissues begin to decay. In the deepest living fishes, this chemical solution comes with trade-offs. The same molecules that protect proteins also affect the chemistry and texture of the body. Pressure adaptation is not a simple strengthening of tissues, but a whole-body biochemical adjustment. TMAO appears to be one important part of that adjustment, alongside changes in membranes, proteins, body density, and the absence of gas-filled spaces. Snailfish of the family Leporidae are the deepest living vertebrates known, with species found at hadal depths exceeding 8,000 meters. The Mariana snailfish, Pseudoliporus swirii, has been recorded from hadal depths close to 8,000 meters in the Mariana Trench, and related snailfish have pushed the observed depth limits for fishes even farther in later expeditions. Snailfish at these depths are pale, translucent, and fragile-looking, with the gelatinous tissue structure that characterizes many deep-sea organisms. Their bodies contain no gas-filled swim bladder, which would be crushed at hadal pressures, relying instead on their overall density being approximately equal to the surrounding water for neutral buoyancy. They feed primarily on small crustaceans called amphipods, which are extraordinarily abundant in hadal trenches because the trenches act as depositional traps for organic materials sinking from above, concentrating food resources at the deepest points of the ocean. This food concentration may explain why hadal environments, despite their extreme conditions, support relatively high animal densities, more abundant life than the surrounding abyssal plain. Amphipods are small crustaceans, superficially resembling shrimp, that are among the most abundant animals in the deep sea and particularly in the hadal zone. In some hadal trenches, amphipods have been found in densities of thousands of individuals per square meter, extraordinary abundance for the deep sea. These hadal amphipods can reach sizes of approximately 30 centimeters, giants compared to the few centimeter amphipods of shallower waters, a phenomenon called deep-sea gigantism that has been observed in several groups of crustaceans and other deep-sea invertebrates. The proposed explanations for deep-sea gigantism include the cold temperatures that slow metabolic rates and may allow continuous growth throughout a long lifespan, the high oxygen concentrations in cold deep water that may support larger body sizes, and reduced predation pressure that removes the size limit imposed by predators in shallower waters. The hadal amphipods descend to the deepest points of the ocean to scavenge on the organic matter that collects there, and they are among the first organisms to arrive at a whale fall or other large food source on the deep seafloor. Whale falls, the bodies of whales that sink to the deep seafloor after death, are one of the most significant episodic food sources in the deep sea. A single large whale carcass can provide an enormous pulse of organic carbon to the otherwise food-limited deep sea floor, sustaining complex biological communities for decades. Whale fall succession follows a characteristic sequence of stages. In the mobile scavenger stage, lasting months to years, large mobile scavengers including sleeper sharks, hagfish, and various large invertebrates consume the soft tissue at rates of up to 40 to 60 kilograms per day. The enrichment opportunist stage follows, lasting months to years, during which dense communities of polychaete worms, crustaceans, and other opportunistic species colonize the exposed bones and surrounding sediment. The sulfophilic stage, which can last for decades in large whale falls, is the most remarkable. Anaerobic bacteria decompose the lipid-rich bone, producing hydrogen sulfide, which supports chemosynthetic communities similar to those at hydrothermal vents and cold seeps. These whale fall communities may include species also found at hydrothermal vents, and whale falls may serve as stepping stones, islands of chemosynthetic habitat scattered across the abyssal plain, allowing vent-associated species to disperse across the ocean floor between the widely separated permanent vent habitats. Hagfish are among the most ancient and most extraordinary living jawless fishes, usually placed with lampreys within the cyclostome branch of vertebrate evolution. Unlike most animals we casually think of as vertebrates, they do not have a true bony backbone in the ordinary sense. They retain a notochord-like support structure rather than the familiar vertebral column. Their lineage reaches far back in evolutionary history, and their body plan preserves features that make them feel like visitors from an older version of the sea. Their anatomy is otherworldly. They have no jaws, no paired fins, and a single nostril. Their eyes are rudimentary structures without lenses or irises that detect only differences in light intensity. They have 4 hearts, and their skin is loosely attached to their bodies, able to move independently in ways that allow them to tie themselves in knots to generate leverage for tearing flesh from a carcass. Most remarkably, hagfish can produce prodigious quantities of slime in response to threat. A single hagfish, when stimulated, can fill a bucket with slime in seconds. The slime is composed of mucin proteins and protein threads, remarkably strong fibers approximately 1 micron in diameter and up to 15 centimeters long, that form a gel when mixed with seawater, clogging the gills of predatory fish and making the hagfish impossible to swallow. This slime defense is so effective that hagfish have very few predators despite living in environments with abundant large predators, and they are found in enormous numbers at whale falls and other food-rich deep-sea sites. The abyssal plain is the flattest terrain on Earth. At depths of 4,000 to 6,000 meters, away from the ridges and trenches, the ocean floor is covered by the soft sediment that has accumulated over millions of years, smoothing and filling any topographic relief in the underlying basaltic crust. The gradient of the abyssal plain is typically less than 1 in 1,000, less steep than an airport runway. This extraordinary flatness, across regions that extend for hundreds to thousands of kilometers without significant topographic feature, is what gives the abyssal plain its name, a vast, level, featureless landscape in the dark. But the appearance of featureless uniformity is deceptive. The abyssal sediment is biologically active, populated by an extraordinary diversity of organisms including polychaete worms, holothurians, foraminifera, nematodes, and various other invertebrates that process the sediment for organic content. The biological communities of the abyssal plain are genuine ecological communities, diverse, structured, and responsive to the sparse but continuous rain of organic particles from above. Sea cucumbers, or holothurians, are among the most abundant visible animals on the abyssal plain, sometimes occurring in densities of hundreds per square meter in organically rich areas. These elongated, soft-bodied animals move slowly across the sediment surface, ingesting sediment and processing it for organic content, essentially acting as the earthworms of the deep seafloor, cycling nutrients through the ecosystem and maintaining sediment structure. Some species can move by releasing their internal organs, including the respiratory trees, intestines, and gonads, through their body walls when threatened, a process called evisceration, and regenerating these organs once the threat has passed. The eviscerated organs may entangle or distract the predator, and the sea cucumber can regenerate a complete set of internal organs within weeks. In some species, evisceration releases sticky Cuvierian tubules, long, sticky threads that entangle predators rather than organs. Deep-sea corals are colonial organisms that build reef structures in the deep ocean, completely independent of the photosynthetic zooxanthellae that provide the primary energy source for tropical shallow water corals. Deep water corals can form extensive reef systems at depths of 200 to 2,000 meters in cold, dark water where photosynthesis is impossible. The primary deep water reef-building corals include Lophelia pertusa and related species, which grow as branching white or pale orange colonies that can form reef mounds tens of meters tall and hundreds of meters in diameter. These reefs are extraordinarily slow-growing. Lophelia grows at approximately 3 to 25 millimeters per year, and radiocarbon dating of deep water coral reefs has found ages of up to 45,000 years for the oldest living corals and even older for fossil reef deposits. Deep water coral reefs support enormous biodiversity, with single reef structures hosting hundreds of species of fish, invertebrates, and other organisms that shelter within and around the complex coral matrix. They are also extremely vulnerable to physical disturbance. Bottom trawling destroys in minutes reef structures that required tens of thousands of years to build. The deepest fish ever filmed was caught on camera in 2023 by scientists from the University of Western Australia and the Tokyo University of Marine Science and Technology, using baited camera landers dropped into the Izu Ogasawara Trench south of Japan. A snailfish was filmed at a depth of approximately 8,336 meters, surpassing the previous depth record for any fish. The fish appeared young and relatively small, moving slowly near the camera bait. This sighting pushed the known depth range of vertebrate life to within approximately 2,000 meters of the deepest point in the ocean, suggesting that the limits of vertebrate life in terms of depth may be deeper than previously estimated, and that even the most extreme hadal environments may support vertebrate life if food resources are available. The ocean at night, at the surface, over deep water far from shore, can display bioluminescence visible to the naked eye. Dinoflagellates, single-celled marine organisms that constitute a significant component of marine plankton, are among the most common sources of sea surface bioluminescence. When mechanically disturbed by a wave, a boat's wake, a fish, or a swimmer, they produce a brief flash of blue-green light. In productive waters where dinoflagellates are abundant, the ocean surface can glow with every movement, waves cresting in blue fire, fish leaving trails of cold blue light, the bow wave of a boat generating a continuous cascade of glowing water. This phenomenon has been observed and described by sailors since antiquity and is documented in the journals of Charles Darwin during the voyage of the Beagle. The flash of bioluminescence produced by a disturbed dinoflagellate is extraordinarily brief, on the order of 100 milliseconds, but in the thousands of individuals disturbed simultaneously by a passing wave, the combined effect is the ocean lighting up. The deep sea contains mineral deposits of potentially enormous economic value. Polymetallic nodules, potato-shaped concretions of iron, manganese, copper, nickel, and cobalt litter the abyssal plain of the Pacific Ocean in particular, having grown by precipitation of dissolved minerals from seawater over tens of millions of years at rates of approximately 10 to 15 millimeters per million years. The nodule fields of the Clarion-Clipperton Zone in the eastern Pacific contain estimated reserves of manganese, cobalt, nickel, and copper vastly exceeding known land-based reserves of these metals, and interest in deep-sea mining of these nodules has grown as demand for metals used in electric vehicle batteries and other clean energy technologies has increased. The ecological consequences of mining nodule fields would be severe. The nodules themselves provide hard substrate for a diverse community of organisms, sponges, corals, worms, crabs, and fish, and the sediment disturbance associated with mining would destroy these communities and create plumes of fine sediment that could spread for hundreds of kilometers. Recovery of biological communities after deep-sea disturbances has been studied through long-term monitoring of sites where sediment was disturbed in experiments decades ago, and the results suggest that recovery is extraordinarily slow. Some disturbed sites show little biological recovery even 40 years after the original disturbance. The giant isopod, Bathynomus giganteus, is one of the most recognizable deep-sea creatures and one of the more dramatic examples of deep-sea gigantism. Related to the common garden woodlouse, the familiar small crustacean that rolls into a ball when disturbed, the giant isopod can reach lengths of up to 45 centimeters and masses of approximately 1.7 kilograms, making it many times larger than any isopod found in shallow water. It lives on the abyssal seafloor at depths of 170 to 2,500 meters, scavenging on dead fish, whales, and other organic matter that sinks from above, as well as predating on slow-moving or sessile organisms. Giant isopods can survive for years without food in the laboratory. Individuals have been documented going without eating for over 4 years while remaining apparently healthy, a testament to their extraordinarily slow metabolic rate. When food does arrive, typically in the form of a large carcass, they can consume enormous amounts rapidly, using their powerful mandibles to tear flesh from even thick-skinned carcasses. The deep sea is one of the most poorly mapped regions of Earth. Current estimates from the GEBCO and Seabed 2030 effort indicate that, as of the 2026 grid release, about 28.7% of the global ocean floor has been mapped to modern standards, meaning that nearly 3/4 of the seabed still lacks detailed modern mapping. The mapping that exists is based primarily on satellite altimetry, measurements of very subtle variations in the height of the ocean surface that reflect the gravitational influence of large seafloor features, which can resolve features a few kilometers across, but misses anything smaller. Direct acoustic mapping using multi-beam sonar from research ships provides much higher resolution, but is extraordinarily slow. A research vessel can survey approximately 2,000 square kilometers per day, and the global ocean floor covers approximately 360 million square kilometers. At that rate, a single ship surveying continuously would take approximately 500 years to map the entire ocean floor. Targeted mapping of specific regions has revealed extraordinary features: submarine volcanoes, landslides, cold seep fields, deep-sea coral reefs, in areas previously assumed to be featureless sediment. The depth at which sunlight is entirely absent, below approximately 1,000 meters in most ocean regions, means that the vast majority of the ocean volume exists in permanent darkness. The euphotic zone, where photosynthesis is possible, extends to approximately 200 meters depth and constitutes approximately 1% of the total ocean volume. The remaining approximately 99% of the ocean volume is aphotic, without sunlight. The organisms that inhabit this vast dark realm depend entirely on organic carbon fixed at the surface and sinking to the deep, or on the chemical energy of hydrothermal and cold seep systems. The biological carbon pump, the process by which surface photosynthesis fixes carbon dioxide into organic matter that sinks to the deep, is one of the most important mechanisms of carbon export from the atmosphere, transferring approximately 5 to 12 gigatons of carbon from the surface to the deep ocean each year. This biological pump is a critical component of the global carbon cycle and helps regulate atmospheric carbon dioxide concentrations on geological and human timescales. Some of the most ancient lineages of life on Earth survive in the deep sea, having changed little from forms that appear in the fossil record hundreds of millions of years ago. The coelacanth, Latimeria, a lobe-finned fish thought to have gone extinct 65 million years ago with the dinosaurs, was discovered alive in 1938 when a specimen was caught in a trawl net off the coast of South Africa. The discovery was one of the most dramatic in the history of zoology, a living fossil from a lineage 400 million years old, closely related to the ancestors of all land vertebrates. The coelacanth has changed little in gross morphology from fossil forms 350 million years old and provides a living window into the body plan of the fish from which tetrapods, four-limbed vertebrates including all amphibians, reptiles, birds, and mammals, evolved. It lives at depths of approximately 150 to 700 meters along the steep, rocky slopes of submarine canyons off eastern Africa and in the waters around Sulawesi in Indonesia. The deep sea may contain the largest population of fish on Earth, despite being the least studied. The mesopelagic zone, between 200 and 1,000 meters depth, is estimated to contain approximately 1 to 10 billion tons of fish biomass, possibly exceeding the total biomass of all commercially exploited fish stocks in the world ocean. These mesopelagic fish, primarily small lanternfish, hatchetfish, and bristlemouth fish typically 2 to 10 centimeters long, are extraordinarily numerous despite being individually tiny. They are important participants in the biological carbon pump through their daily vertical migration, ascending to the surface at night to feed on zooplankton and descending to the mesopelagic during the day, transporting carbon fixed at the surface to depth in their bodies and through respiration. The potential for commercial exploitation of mesopelagic fish, driven by demand for fish meal and fish oil for aquaculture feed, is a growing concern in fisheries science, and the ecological consequences of large-scale harvest from this little-studied ecosystem are essentially unknown. The deep sea also changes the meaning of color. At the surface, color is a world of reflection: red flowers, green leaves, blue sky, the familiar palette of light bouncing from objects into the eye. Underwater, color disappears with depth because water absorbs longer wavelengths first. Red vanishes within the first few meters. Orange and yellow fade soon after. Blue penetrates deepest, which is why the open ocean looks blue and why most deep-sea bioluminescence is blue-green. In a world where red light is absent, a red animal does not appear bright. It appears black. This is why many mesopelagic animals are colored red or deep burgundy, even though no red light reaches them. What would be conspicuous in a tide pool becomes a form of invisibility in the twilight zone. Counterillumination is one of the most elegant camouflage strategies in the ocean. Many animals living in the mesopelagic zone have rows of light-producing organs along their undersides. To a predator looking upward, the animal's body would normally appear as a dark silhouette against the faint light filtering down from above. By producing light from below at the same brightness and color as the background, the animal erases its own shadow. It does not hide by becoming dark. It hides by becoming the same faint blue as the water above it. Counterillumination requires a remarkable degree of control because the brightness of the downwelling light changes with depth, time of day, cloud cover, moonlight, and water clarity. The animal must match not a fixed color, but a shifting field of dimness. The daily vertical migration of deep-sea animals is the largest migration on Earth by biomass, and it happens every night. As the sun sets, enormous numbers of small fish, squid, crustaceans, and gelatinous animals rise from the mesopelagic zone toward the surface waters to feed under the cover of darkness. Before dawn, they descend again, often hundreds of meters, to avoid visual predators during the day. This movement is so massive that sonar instruments during the Second World War detected it as a false seafloor, a scattering layer that rose and fell each day. What looked like a geological boundary was actually life, billions upon billions of small bodies moving in coordinated response to light. The ocean itself seemed to breathe upward at night and downward at morning. This migration is also a major part of Earth's carbon cycle. When mesopelagic animals feed near the surface at night, they consume carbon-rich organic matter produced by photosynthesis. When they descend during the day, they carry some of that carbon with them into deeper water, where it may be respired, excreted, or eventually sink farther toward the seafloor. This active transport, combined with the passive sinking of marine snow, is part of the biological carbon pump, the process by which the ocean moves carbon from the atmosphere-connected surface into the deep ocean. It is quiet, continuous, and globally significant. Some of the carbon in the deep sea is locked away from the atmosphere for decades, centuries, or longer. The darkness below helps regulate the air above. Marine snow is not snow in the ordinary sense, but it behaves like weather in the deep ocean. It is a slow fall of organic fragments, dead plankton, mucus, fecal pellets, tiny shells, bacterial aggregates, fragments of jellyfish, bits of animals, bits of plants, and mineral particles. Under strong lights from a submersible, it drifts through the water like pale flakes in a winter night. Most of it is consumed before it ever reaches the bottom. Bacteria break it down. Zooplankton eat it. Filter feeders catch it. Only a small fraction survives the long descent to the abyss. But over the huge area of the ocean floor, even that small fraction matters. It is the main food supply for much of the deep sea, a thin rain of surface life falling into darkness. Gelatinous animals are among the most important and most underappreciated inhabitants of the deep ocean. Jellyfish, siphonophores, comb jellies, salps, larvaceans, and many other transparent or translucent animals drift through the water column in forms that often collapse when collected in nets. For a long time, traditional sampling methods underestimated them because soft bodies were torn apart before reaching the surface. Only with remotely operated vehicles, gentle samplers, and high-resolution underwater cameras did researchers begin to appreciate how abundant and diverse these animals are. Some are predators. Some are filter feeders. Some build mucus houses that concentrate particles from the water. Their bodies are mostly water, but their ecological role is not small. They are part of the ocean's hidden architecture. Siphonophores are especially strange because what looks like one animal is actually a colony of specialized individuals functioning together as a single organism. The Portuguese man of war at the surface is the familiar example, but deep-sea siphonophores can be far more spectacular. Some form long, delicate chains of repeated units, stretching tens of meters through the water. Each unit has a role: floating, swimming, feeding, reproducing, capturing prey. The colony functions as one, but it is built from many. In the deep sea, where food may be rare and prey may appear unpredictably, a long drifting net of stinging tentacles can be an effective solution. The animal becomes less like a creature moving through a world and more like a living trap suspended in water. The giant siphonophore discovered in deep waters off Western Australia in 2020 was estimated to form a spiral colony perhaps more than 40 meters long, making it one of the longest animals ever observed. It was not a whale, not a shark, not a fish with bones and muscle, but a fragile floating colony whose body would have been destroyed by a net. Its discovery was a reminder that many of the largest organisms in the ocean are not large in the way land animals are large. They are long, transparent, modular, and almost impossible to preserve. Their size is not obvious from a museum specimen. It has to be seen alive, in place, in the water column for which it evolved. The deep sea has its own version of forests. Deep water coral reefs grow in cold darkness, without the symbiotic algae that fueled tropical reef-building corals in shallow sunlight. These corals capture plankton and organic particles from passing currents, building branching skeletons slowly over centuries or millennia. Some deep coral structures are older than recorded human history. They provide habitat for fish, crustaceans, brittle stars, sponges, worms, and many other organisms. To a camera descending into the dark, a deep coral reef can appear suddenly out of empty water. Pale branches, orange fans, white cups, soft polyps open to the current. It is not a tropical reef, but it is a reef nonetheless, a living structure built in darkness. Glass sponges form another kind of deep-sea architecture. Their skeletons are made of silica, the same basic material as glass, arranged into intricate frameworks of spicules. Some species form reefs, especially in cold waters of the North Pacific, where sponge skeletons accumulate into complex, three-dimensional structures. These sponge reefs can support rich communities of animals, but they are fragile. Their skeletons break easily under physical disturbance, and because many deep-sea organisms grow slowly, damaged communities may require decades, centuries, or longer to recover. The deep sea often gives the illusion of stability because change is slow. But slow recovery does not mean resilience. It can mean that damage persists for longer than a human lifetime. The deep ocean is not silent. Sound travels farther in seawater than light, and many marine animals use sound to communicate, navigate, or detect their environment. Low-frequency sounds can travel across enormous distances, especially through layers such as the SOFA or channel, where sound becomes trapped and guided by the structure of temperature and pressure in the water column. Whales use this property of the ocean to communicate across distances that would be impossible in air. Human activity has added new sounds to this world: ship engines, sonar, seismic surveys, construction, and explosions. For animals adapted to a naturally dark environment where sound may carry crucial information, noise pollution is not a small disturbance. It changes the acoustic landscape. The deep sea is also chemically layered. Oxygen is not evenly distributed through the water column. In many tropical and subtropical regions, an oxygen minimum zone forms at intermediate depths, where oxygen is consumed by the decay of sinking organic matter faster than it is replenished by mixing. Some animals avoid these low-oxygen waters. Others specialize in them. The vampire squid is one of the most famous examples, a slow, efficient animal living in a region where many predators cannot function well. In oxygen-poor water, speed is expensive and often impossible. Survival favors stillness, efficiency, patience, and the ability to make a living from small particles drifting through the dark. Oxygen minimum zones are expanding in some parts of the ocean as warming reduces oxygen solubility and changes stratification. This is one reason deep-sea science is not only about strange animals and distant trenches. The deep ocean is part of Earth's climate system, and its chemistry is changing. When oxygen declines, habitats shift. Animals move if they can, adapt if they can, or disappear from areas where their metabolism can no longer function. The deep sea may look remote, but it is connected to surface warming, nutrient cycles, carbon dioxide, and the long-term health of marine ecosystems. The darkness is not separate from the world above it. Brine pools are among the strangest seafloor environments. They occur where extremely salty water collects in depressions on the seafloor, forming a dense liquid that sits beneath ordinary seawater like a lake beneath the sea. The boundary between the brine and the surrounding water can look like a shimmering surface. Submersible lights reveal ripples, shorelines, and even waves along the edge of the pool. To many animals, the brine is toxic or suffocating because it may contain very little oxygen and high concentrations of methane or hydrogen sulfide. But specialized microbial communities can live at the margins, where chemical gradients provide energy. A brine pool is one of those places where the seafloor begins to look like another planet. Hydrothermal vents also create mineral landscapes that can grow, collapse, and rebuild over time. Chimneys may form rapidly as minerals precipitate from hot fluid meeting cold seawater. A vent that is active for years may host dense life. Then the plumbing below the seafloor can shift, the vent may slow or stop, and the community, dependent on its chemistry, may decline. A new vent may open elsewhere. The deep sea is often imagined as still, but hydrothermal systems are dynamic, changing over years and decades. Earthquakes, volcanic eruptions, and tectonic movement reorganize them. The seafloor is not a dead floor beneath the ocean. It is part of a restless planet. At vents and seeps, animals often live by outsourcing metabolism to microbes. Mussels and clams can host chemosynthetic bacteria in their tissues. Shrimp may graze bacterial films from rock surfaces. Tube worms rely on internal bacterial partners. Some snails and limpets feed on microbial mats. These systems reverse the familiar surface world hierarchy. On land and in shallow seas, plants and algae capture sunlight, and animals eat them or eat each other. At vents, bacteria and archaea capture chemical energy, and animals build their lives around them. The base of the food web is not green. It is microbial, chemical, and often invisible. The scaly foot snail, discovered at hydrothermal vents in the Indian Ocean, looks almost mythical. Its foot is covered with hard scales containing iron sulfide minerals, giving it a dark metallic armor. Its shell also contains iron compounds. The snail lives in hot, chemically extreme vent fields, hosting symbiotic bacteria that help provide nutrition. It is one of the few known animals to incorporate iron sulfides into protective body structures. Its existence seems like something from speculative fiction, a snail in armor, living beside black smokers, built partly from the chemistry of the vent itself. Yet it is real, vulnerable, and known from only a small number of vent fields. Yeti crabs, first described in the early 21st century, are another example of how recently the deep sea continues to reveal major new animal forms. These pale crustaceans have hairy-looking arms covered with setae that host bacteria. In some species, the crabs wave their arms in vent fluids, apparently farming or encouraging bacterial growth, which they then consume. Their bodies are adapted to narrow thermal and chemical zones. Too far from the vent, and the food source fades. Too close, and the water may be too hot or chemically harsh. Life at vents often means living on a boundary, balancing between cold ocean and hot earth, oxygen and sulfide, safety and energy. Cold seeps have their own slow drama. Methane bubbles rise from sediments. Bacterial mats spread in white patches. Clams and mussels cluster in beds. Tube worms may live for centuries, extending roots into the sediment to access sulfide, while their plumes reach into oxygenated water. Compared with hydrothermal vents, cold seeps can be stable for much longer periods, and the animals that live there often grow slowly. A seep community may persist through many human generations, quietly supported by fluids rising from buried organic matter, gas hydrates, or deeper geological reservoirs. It is not dramatic like a smoking chimney, but it is equally alien in its dependence on chemistry from below. Methane hydrates make the seafloor feel almost paradoxical. They are ice-like, but they can burn when brought to the surface and ignited because methane is trapped inside cages of water molecules. In the deep sea, they are stable because of high pressure and low temperature. They represent a large store of carbon, but not a simple one. Some methane is consumed by microbes before it escapes. Some supports seep ecosystems. Some may be destabilized by warming, though the rate and scale of that process vary by region. Methane hydrate is a reminder that the seafloor is not only biology and rock. It is chemistry held in delicate balance by pressure, temperature, and time. The deep sea also contains life inside the seafloor, not just above it. Beneath the sediments and crust, microbial communities live in pores, cracks, and chemically active zones. Some of these microbes have extremely slow metabolisms. They may divide rarely, perhaps over years, decades, or longer, surviving on tiny amounts of energy from chemical reactions in rock and sediment. The deep biosphere may contain a substantial fraction of Earth's microbial life. It is one of the strangest ideas in modern biology that beneath the visible world, beneath the ocean floor, there is another living world, mostly microbial, slow beyond ordinary imagination, powered not by sunlight, but by chemistry and the faint persistence of metabolism. Time behaves differently in the deep sea because many processes are slow. Sediment accumulates slowly. Nodules grow slowly. Corals grow slowly. Some animals mature slowly and live for decades or centuries. A trawl can destroy a coral garden in minutes that took thousands of years to build. A mining test can leave marks still visible after decades. A whale fall can feed a community for years. A deep-sea clam may live long enough that the world at the surface changes politically, technologically, and culturally, while the clam remains in its dark chemical habitat. The deep sea teaches a different sense of duration, one less connected to human schedules. Orange roughy is a deep-sea fish famous not because of its appearance, but because of the lesson it taught fisheries science. It grows slowly, matures late, and can live for more than a century. When commercial fisheries began harvesting it heavily, populations could not recover quickly because the species was not biologically built for rapid replacement. Many deep-sea fish share this vulnerability. The traits that allow survival in a cold, food-poor environment, slow growth, long life, delayed reproduction, low energy use, make them poorly suited to heavy exploitation. A fish that lives slowly cannot be harvested as if it lived fast. The Greenland shark, although not a classic abyssal animal, belongs to the cold, dark world of northern deep waters and may be the longest-lived vertebrate known. Studies using radiocarbon dating of islands proteins suggest that some individuals may live for centuries, possibly around 400 years. They grow slowly, move slowly, mature late, and inhabit a world where cold temperatures reduce metabolic speed. A shark alive today may have been born before the Industrial Revolution. The idea is difficult to hold in the mind. A large animal swimming quietly through Arctic depths, carrying within its tissues a timeline longer than many human nations. Deep-sea animals often look strange because they are built for scarcity. Large mouths, expandable stomachs, long teeth, lures, slow metabolism, transparent tissues, delicate bones, and patient behavior are not random weirdness. They are solutions to an environment where energy is rare and encounters are unpredictable. A fish that meets prey only occasionally must not miss the chance. A predator may need to swallow something almost as large as itself. A scavenger must be able to eat heavily when food appears and survive long periods without it. The deep sea rewards exaggeration of certain traits because ordinary surface world proportions are not always useful in darkness and hunger. Bristlemouth fish may be the most abundant vertebrates on Earth. They are small, dark, and easily overlooked, living in the mesopelagic zone in enormous numbers. Some estimates suggest they may number in the trillions or more, far exceeding humans, birds, mammals, and perhaps most other vertebrate groups. They are not famous like sharks or whales because they are small and live far below casual observation, but their collective ecological importance is enormous. They participate in vertical migration, carbon transport, and deep-water food webs. The most common vertebrate on Earth may be one that most people have never heard of and will never see alive. Lanternfish are similarly important. They have rows of photophores along their bodies, often arranged in species-specific patterns that may help individuals recognize one another in the dark. They rise at night, descend by day, and form dense acoustic scattering layers detected by sonar. They are food for squid, larger fish, seabirds, seals, whales, and many predators that connect surface ecosystems to deeper ones. Because they are so abundant, there has been interest in harvesting mesopelagic fish for fish meal and oil, especially for aquaculture. But scientists remain cautious because the mesopelagic zone is poorly understood, and removing large amounts of biomass from the ocean's midwater could alter carbon cycling and food webs in ways that are difficult to predict. The hatchetfish has a body shaped like a small silver blade and eyes that look upward. Its thin body, reflective sides, and ventral photophores help it vanish in the dim water. From the side, its reflective body may mirror the faint environment. From below, its counterillumination reduces the silhouette. The shape is not ornamental. It is a carefully tuned optical solution to a world where being seen can mean being eaten. In the twilight zone, survival is often a matter of angles. What a predator sees from below, what prey sees from above, how light enters the water, how a body breaks its outline, and how brightness can be adjusted to match the fading sky. The gulper eel, sometimes called the pelican eel, has an enormous mouth attached to a long, slender body. Its mouth can open wide like a net, but its body is not built like that of a powerful, fast predator. It likely feeds on small crustaceans and other animals, using its large mouth to scoop prey in an environment where food is sparse. Its tail can produce light, possibly used in communication or prey attraction. Like many deep-sea animals, it became famous because it looks almost impossible, but its appearance makes sense when seen as an energy-saving design. A large capture surface, a slender body, and a life adapted to rare opportunities. The tripod fish rests on the seafloor using elongated fin raised like stilts. It positions itself facing into the current, lifting its body above the sediment while its long pectoral fins extend outward like antennae. In the darkness, with little need for rapid swimming, it waits for small crustaceans drifting past in the current. Its posture is so still that it can look more like equipment than an animal. But it is a living solution to abyssal feeding. Let the current bring food, minimize energy use, and use sensitive fins to detect prey. It is patience made anatomical. The deep sea is full of animals that blur the line between plant-like stillness and animal life. Sea pins stand upright in soft sediment, their polyps feeding from the current. Glass sponges filter water through delicate skeletons. Anemones cling to rocks, whale bones, or artificial structures. Crinoids extend feathery arms into the flow. These organisms do not chase food. They wade in the current and make the ocean do some of the work. In a food-poor world, stillness can be strategy. Movement costs energy. Posture, placement, and patience can save it. Some deep-sea animals are almost transparent. Transparency is a form of camouflage in open water where there is no rock, seaweed, or sand to hide against. A transparent body lets light pass through it, reducing the animal's visibility. But transparency has limits. Eyes, digestive organs, and reproductive tissues often cannot be made fully transparent, so some animals minimize, mirror, or hide these structures. The glass squid, glass octopus, and many gelatinous animals show variations on this theme. In the deep pelagic ocean, invisibility is not blackness. Sometimes it is the absence of obvious body at all. The deep sea also preserves records of catastrophe. Turbidites, layers of sediment deposited by underwater landslides and gravity-driven flows, can spread across abyssal plains after earthquakes or slope failures. These layers can be recognized in sediment cores and used to reconstruct past seismic events. The ocean floor keeps a memory of shaking continents, collapsing slopes, volcanic eruptions, and ancient climate shifts. Its quiet surface of sediment is not blank. It is layered history. Each thin band may represent a change in circulation, a volcanic ash fall, a bloom at the surface, a glacial cycle, or a sudden disturbance that traveled through the deep as a dense cloud of suspended sediment. Submarine canyons cut through continental margins like underwater river valleys. Some begin near the edge of continental shelves and descend thousands of meters toward the deep ocean. They funnel sediment, organic matter, and sometimes pollutants from shallow waters into the deep sea. Currents can be accelerated through them, creating habitats richer in food than the surrounding slope. Many deep-sea animals gather in and around submarine canyons because the supply of particles from the surface and shelf is higher there. A canyon is a corridor between worlds, the productive shallow sea above and the dark, deep ocean below. Tsunamis also connect the deep ocean to human life, though not in the way ordinary waves do. In the open ocean, a tsunami may have a low height and an enormous wavelength, traveling at jet aircraft speeds across the deep sea with little visible disturbance at the surface. Its speed depends on water depth. The deeper the ocean, the faster the wave travels. When it approaches shallow coastal water, it slows, compresses, and grows in height, becoming dangerous. The deep sea, therefore, is part of the hidden physics of coastal disasters. The shape of the seafloor influences how energy moves toward shore. The ocean floor contains transform faults, fracture zones, volcanic plateaus, trenches, ridges, abyssal hills, and seamount chains. Many of these features are known only from sonar maps and satellite-derived gravity data. A seamount may rise thousands of meters from the seafloor and still never reach the surface. Around it, currents accelerate and lift nutrient-rich water, creating biological hotspots. Fish aggregate there. Corals and sponges may attach to hard rock. Predators visit. Some seamount species are endemic, found only on one mountain or one chain of mountains. A seamount is an island that never breaks the surface, isolated not by air but by depth. The mid-ocean ridge system is not only the longest mountain chain on Earth, it is also the place where the planet continually makes new seafloor. As plates pull apart, mantle material rises, melts, and forms basaltic crust. This new crust records the direction of Earth's magnetic field as it cools. On either side of ridges, alternating bands of magnetic polarity preserve the history of geomagnetic reversals, like a tape recording written into rock. The recognition of these symmetrical stripes helped confirm plate tectonics, one of the great scientific revolutions of the 20th century. The deep ocean floor helped reveal that Earth itself is mobile. The age of the ocean floor is surprisingly young compared with the continents. Most oceanic crust is less than 200 million years old because it is continually created at mid-ocean ridges and destroyed at subduction zones. Continental rocks can be billions of years old, but oceanic crust is recycled into the mantle. The seafloor is not a permanent floor. It is a moving surface, born, carried, altered by sediment and life, then eventually pulled back into Earth. The ocean covers most of the planet, but the floor beneath it is part of a geological conveyor belt. Deep ocean circulation links distant parts of the planet. Cold, dense water forms near the poles, sinks, and spreads through the deep basins. This overturning circulation transports heat, oxygen, nutrients, and carbon around the globe. A parcel of deep water may remain away from the surface for hundreds or even more than 1,000 years before rising again. The deep ocean is therefore a reservoir of memory. It stores the chemical signature of past atmospheric conditions, the temperature of polar regions, and the carbon absorbed at the surface. When scientists measure deep water properties, they are not only measuring the present. They are reading the slow movement of past climate through the ocean. The deep sea has been explored by people only in fragments. In 1960, Jacques Picard and Don Walsh descended in the bathyscaphe Trieste to the Challenger Deep, becoming the first humans to reach the deepest known point of the ocean. In 2012, James Cameron made a solo descent in Deep-Sea Challenger. In later years, full ocean depth submersibles and landers expanded sampling and observation. But the number of people who have personally visited the deepest seafloor remains tiny compared with the number who have been to space. The deep ocean is not far away in cosmic terms. It is on this planet. But technically, psychologically, and physically, it remains remote. Submersibles are only one part of modern deep-sea exploration. Remotely operated vehicles, connected to research vessels by long cables, can descend for hours, transmit high-definition video, use robotic arms, collect samples, and place instruments. Autonomous underwater vehicles can survey large areas without a human pilot, following programmed paths and using sonar, cameras, chemical sensors, and mapping systems. Baited landers can sit on the seafloor and wait for animals to arrive. Moorings can measure temperature, currents, oxygen, and sound for months or years. Each tool sees a different part of the deep sea. No single instrument can capture the whole. One reason the deep sea remained misunderstood for so long is that bringing animals to the surface changes them. Pressure drops, temperature changes, delicate tissues tear, transparent bodies collapse, gas-filled spaces expand, colors change under daylight, behaviors stop. An animal seen dead in a jar may reveal anatomy, but not life. It took cameras in the deep ocean to show how these animals actually move, feed, glow, hide, and interact. Many deep-sea creatures that looked grotesque in early specimens appear graceful when observed alive in their own water, under the pressures and darkness for which they are built. The barrel-eye fish is a perfect example of this. For decades, specimens brought up in nets were damaged, and the transparent dome over the head was usually destroyed. Only when living barrel eyes were filmed in place did scientists see the fluid-filled shield and the rotating tubular eyes. The animal had not been fully understood because it could not survive ordinary collection. The deep sea often hides its secrets not because they are mystical, but because our methods damage the very things we are trying to understand. Observation has to become gentler before knowledge becomes accurate. The same is true for behavior. A deep-sea squid preserved in a museum cannot show hunting, escape displays, mating, camouflage, or bioluminescent signaling. A fish specimen cannot show how it holds position in the current. A jellyfish torn by a net cannot show the architecture of its tentacles. Many discoveries in modern deep-sea biology are not new species, but new behaviors. An animal using its light in an unexpected way, a squid carrying eggs, a fish walking on the bottom, an octopus brooding longer than any animal known, a crab farming bacteria, a sponge reef hosting a community. Life is not only anatomy, it is action. The deep sea contains many species still undescribed by science. Every expedition to poorly explored regions has the potential to find organisms unknown to formal taxonomy. Some are tiny worms or crustaceans. Some are microbes. Some are new fishes, corals, sponges, or gelatinous animals. The challenge is not only finding them, but describing them, sequencing them, comparing them, understanding their ecology, and placing them within the tree of life. A new species name is only the beginning. The larger questions are where it lives, what it eats, how it reproduces, how long it lives, what threatens it, and what it can tell us about evolution. Reproduction in the deep sea often reflects the difficulty of finding mates in a vast, dark, low-density environment. Some animals release eggs and sperm into the water and rely on timing, currents, or aggregation. Some carry eggs. Some produce enormous numbers of offspring because most will never survive. Some deep-sea anglerfish evolved permanent attachment between male and female, a solution so extreme it can sound impossible until the ecological problem is understood. In an environment where a suitable mate may be rare, the first successful encounter may need to become permanent. Reproductive biology in the deep sea is often biology shaped by loneliness. Brooding can be extraordinarily costly in the deep ocean. The deep-sea octopus Granulidon boreopacifica was observed guarding eggs for more than 4 years, the longest known brooding period for any animal. During that time, the female stayed with her eggs, protecting and ventilating them, while her body slowly deteriorated. The cold, deep water slows embryonic development, stretching parental care across a timescale almost unimaginable for an animal whose shallow water relatives may live only a year or two. In the deep sea, even motherhood can become a long vigil in the dark. Deep sea gigantism appears in several animal groups: giant isopods, giant amphipods, large sea spiders, large squid, and other invertebrates. The causes are not simple and probably differ by group. Cold temperatures can slow metabolism and extend lifespan, allowing continued growth. High pressure may influence physiology. Food scarcity may favor larger size and scavengers able to travel farther or store more energy. Reduced predation may allow larger bodies. Higher oxygen content in cold water may support larger animals in some cases. The pattern is real, but it is not one single rule. The deep sea does not make everything giant. It makes some lineages giant under certain conditions for reasons still being studied. The colossal squid, living in cold Southern Ocean depths, reminds us that some of the ocean's largest predators are rarely seen alive. It has massive eyes, powerful arms and tentacles, and rotating hooks on some of its limbs. Much of what is known comes from specimens found in the stomachs of sperm whales or captured accidentally by fisheries. Sperm whales dive into deep water to hunt squid, and the circular scars on their skin show encounters with large cephalopods. The deep sea is not empty space between surface and floor. It is an arena of predator and prey, much of it never witnessed directly by humans. Sperm whales themselves are surface-breathing animals that enter the deep sea as visitors. They can dive more than a kilometer, sometimes deeper, holding their breath while hunting squid and fish in darkness. Their echolocation clicks are among the loudest biological sounds produced by any animal. The clicks help them navigate and locate prey in a world where vision is limited. A whale descending from sunlight into black water carries air from the surface into the deep, then returns to breathe again. It links two worlds with every dive. Elephant seals do something similar. They can spend months at sea, diving repeatedly into the mesopelagic and bathopelagic zones to feed. Some dives exceed 1,000 meters. Instruments attached to seals have become valuable scientific tools, measuring temperature and salinity in regions difficult for ships to sample, especially around Antarctica. The animal's own life becomes a path through data. While it hunts in darkness, it also records the structure of the ocean. Deep-sea science sometimes depends on machines. Sometimes it depends on animals carrying instruments into places people rarely reach. The deep sea is connected to the surface through falling bodies. When a large fish dies, when a squid sinks, when a balloon collapses, when fecal pellets drop, when a whale carcass descends, energy moves downward. A whale fall is the most dramatic example, but smaller falls happen constantly. The deep sea is fed by death and leftovers from above. This might sound bleak, but ecologically it is beautiful. Nothing is simply wasted. Matter moves. Energy is transformed. A body that once swam in sunlight becomes food for hagfish, worms, bacteria, clams, and communities that may last for decades. Death at the surface becomes life in the dark. Even wood can create temporary deep-sea ecosystems. Logs, branches, and plant material washed into the ocean can sink to the seafloor, where would boring bivalves, bacteria, worms, and other organisms colonize them. Wood falls may have served as evolutionary stepping stones for animals that later adapted to whale falls, seeps, and vents. The idea is simple and elegant. Isolated patches of concentrated organic matter scattered across the seafloor create islands of opportunity. Animals able to find and exploit these islands may disperse from one to another across the deep ocean. Shipwrecks can also become artificial deep-sea habitats. Metal hulls, wooden structures, cargo, and other hard surfaces provide substrate in areas where the natural seafloor may be soft sediment. Over time, microbes, corals, sponges, anemones, fish, and crustaceans may colonize them. A wreck is a human object transformed by the ocean into habitat and history at once. In shallow water, wrecks are often visited by divers. In the deep sea, they become quiet archaeological and ecological sites, slowly corroding, slowly accumulating life, often undisturbed for decades. The Titanic, resting at approximately 3,800 meters in the North Atlantic, is one of the most famous deep-sea wrecks. Its slow decay is driven by corrosion, currents, and microbial activity, including rusticle-forming bacteria that consume iron and produce icicle-like formations of corrosion products. The wreck is both a memorial and an ecosystem. It is not eternal. The deep sea preserves some things by cold and darkness, but it also transforms them. Steel becomes rust. Wood may disappear. Objects become habitat. Human history enters geological and biological time. Microplastics have now been found even in the deepest parts of the ocean. Fibers and fragments from human materials can travel through rivers, atmosphere, surface waters, sinking particles, and animal bodies, eventually reaching trenches and abyssal sediments. The presence of plastics in the deep sea shows that remoteness is not protection. A place can be unvisited by humans and still receive human material. This is one of the unsettling lessons of modern ocean science. The deep sea is not outside civilization's reach. It is affected by what happens at the surface, even when no one is watching. Chemical pollutants also reach deep environments. Persistent organic pollutants, heavy metals, and industrial compounds can bind to particles, enter food webs, and accumulate in long-lived animals. Some hadal amphipods have been found with surprisingly high levels of human-made pollutants, despite living in trenches far from direct industrial sources. The ocean connects everything through currents, sinking matter, and food chains. Distance dilutes some impacts, but not all. The deepest places on Earth are not isolated vaults. They are part of a planetary circulation system. Climate change is altering the deep ocean slowly but measurably. Heat absorbed at the surface can be transported downward by mixing and circulation. Oxygen levels can shift. Acidification changes carbonate chemistry, affecting organisms that build shells and skeletons from calcium carbonate. Food supply to the deep may change as surface productivity changes. Deep-sea ecosystems often respond slowly, but slow response does not mean no response. The deep ocean is vast, but not immune. Its stability is one of the things that makes change there so important. Deep-sea mining is one of the most controversial questions in ocean policy. Polymetallic nodules contain metals used in batteries and electronics, including nickel, cobalt, copper, and manganese. Supporters argue that these metals are needed for energy transitions and that seafloor mining may reduce pressure on some land-based mines. Critics argue that the ecosystems of nodule fields are poorly understood, recovery is extremely slow, sediment plumes could spread widely, and species may be lost before they are even described. The debate is difficult because it sits at the intersection of climate technology, biodiversity, economics, law, and uncertainty. What is clear is that the deep seafloor is not empty. The nodules are habitat, not just ore. Polymetallic nodules grow at rates so slow they challenge ordinary economic imagination, often millimeters over millions of years. A nodule lifted from the seafloor may have been forming since before the rise of humans, before the ice ages, before many modern species existed. On and around it, small animals live, attach, hide, feed, and reproduce. To a mining machine, it may look like a resource. To an organism, it may be the only hard surface in a landscape of soft sediment. The same object can be geology, chemistry, habitat, and commodity, depending on who is looking. The deep sea also matters for medicine and biotechnology. Enzymes from heat-loving microbes helped make modern molecular biology possible, because heat-stable polymerases allow repeated heating cycles in PCR. Other deep-sea organisms produce unusual molecules that may have pharmaceutical or industrial value. Extreme environments select for unusual chemistry. Proteins stable under pressure, enzymes active at high temperature, compounds used in defense, communication, or symbiosis. This does not mean the deep sea is simply a storehouse for human use. It means that life there has solved biochemical problems very different from those at the surface, and those solutions can teach us. Hydrothermal vents also changed the search for life beyond Earth. Before their discovery, sunlight seemed almost inseparable from complex ecosystems. Vents proved that chemical energy alone could support rich communities. That shifted how scientists think about icy moons such as Europa and Enceladus, where liquid oceans may exist beneath ice shells and where water rock chemistry could provide energy. The deep ocean became an analog for worlds far from the sun. Studying Earth's vents is not only oceanography. It is astrobiology, a way of asking whether life could exist in dark oceans elsewhere. Europa, a moon of Jupiter, likely has a global ocean beneath its icy crust. Enceladus, a moon of Saturn, ejects plumes of water vapor and ice grains from fractures near its south pole, allowing spacecraft to sample material that may come from a subsurface ocean. If hydrothermal activity occurs on the rocky seafloors of these moons, chemical energy could be available. No one knows whether life exists there. But the deep sea made the question scientifically serious. A dark ocean is no longer automatically a dead ocean. On Earth, darkness did not prevent life from becoming strange, complex, and abundant. The deepest places in Earth's ocean are not only cold and dark, but geographically narrow. Trenches form where one tectonic plate bends and descends beneath another. They are long, curved, steep-sided depressions, not open basins. Their shape affects currents, sediment transport, food accumulation, and animal communities. Organic matter can slide, fall, or be funneled into trenches, making some hadal environments richer in food than the abyssal plains around them. This may help explain why amphipods and snailfish can be relatively abundant in places that seem, from the surface, impossibly hostile. The trench is both extreme and, in some ways, concentrated. Pressure limits life in subtle ways. It affects protein folding, membrane fluidity, enzyme reactions, and the behavior of water itself. Deep-sea organisms do not survive pressure by resisting it like a submarine hull. Their bodies are mostly water and are not crushed because there are few compressible gas spaces. The challenge is biochemical. Membranes must stay flexible. Proteins must keep their shape. Molecular machines must operate under conditions that would destabilize surface-adapted cells. Pressure adaptation is therefore an invisible architecture written into lipids, proteins, solutes, and cellular regulation. TMAO is one part of that adaptation in many deep-living fishes, but it also appears to set limits. At extreme depths, the concentration needed to counteract pressure may itself become physiologically costly. Some researchers have proposed that this chemical constraint helps explain why fish have not been found at the very deepest point of the ocean, even though invertebrates and microbes occur there. The deepest vertebrates live remarkably deep, but perhaps not infinitely deep. Life reaches toward the bottom and then meets a biochemical boundary. The Mariana snailfish and other hadal snailfishes are not armored monsters. They look delicate, pale, soft, and almost translucent. Their bodies are adapted to pressure not by hardness, but by flexibility and chemistry. They feed on amphipods, move slowly, and live in a world with no sunlight and crushing pressure. Their existence is surprising because vertebrates are often associated with more moderate environments. Yet here are fish near the limits of vertebrate life, not fighting the trench with strength, but belonging to it. At the bottom of trenches, human objects have also arrived. Landers, sampling gear, submersibles, and unfortunately sometimes trash have reached hadal depths. Plastic bags and other debris have been observed in deep trenches. These sightings are jarring because the trench seems like the ultimate remote place, and yet the signature of the surface is there. The deepest point of the ocean is not beyond human influence. It is part of the same planet, connected by gravity, currents, materials, and time. The abyssal plain, though often described as featureless, is full of small-scale structure. Tracks from crawling animals cross the sediment. Burrows open into the mud. Holothurians leave feeding trails. Tiny mounds and depressions mark biological activity. Under a camera's light, the plain becomes less empty. Brittle stars, sea cucumbers, worms, sponges, small fish, crustaceans, and single-celled organisms appear. The animals may be sparse compared with a coral reef, but they are present, diverse, and adapted to a landscape where each particle of food matters. Emptiness is often just a limitation of human attention. Xenophyophores are giant single-celled organisms found on the deep seafloor, some reaching sizes of several centimeters or more. They build delicate nests from sediment particles and organic material, creating structures that can provide habitat for small animals. The idea of a single cell large enough to be seen easily, shaping sediment into a home, challenges the ordinary intuition that single-celled life must be microscopic and simple. In the deep sea, even the boundary between microbial and visible worlds becomes less straightforward. Foraminifera, relatives of xenophyophores, leave shells that accumulate in deep-sea sediments. Their tiny tests preserve chemical signatures of the water in which they formed. By analyzing oxygen isotopes and other signals in fossil foraminifera, scientists reconstruct past ocean temperatures, ice volume, circulation, and climate. Much of what we know about Earth's glacial cycles comes from these small shells in deep sediment cores. A microscopic organism sinks, becomes part of the seafloor, and millions of years later helps reveal the climate history of the planet. Sediment cores are time machines. A cylinder of mud retrieved from the deep ocean can contain layer after layer of past environments. Volcanic ash marks eruptions. Microfossils show which organisms lived at the surface. Chemical ratios show temperature and ice volume. Dust from continents shows wind patterns and aridity. Organic molecules show productivity and oxygen conditions. The deeper the core, the farther back in time it reaches. Those sediments can be disturbed, mixed, or missing depending on location. The deep sea preserves history not in written language, but in particles. The darkness of the deep ocean also changes how animals use vision. Eyes may become larger, more sensitive, tubular, or reduced, depending on depth and lifestyle. In the twilight zone, where faint light still exists, enormous eyes can detect silhouettes and flashes. In complete darkness, vision may be used mainly for bioluminescent signals. In caves or deep sediments, eyes may become less important. Evolution does not simply make eyes bigger in the dark. It shapes them according to what kind of light exists, what information that light carries, and whether the cost of maintaining vision is worth paying. Some deep-sea animals use mirrors. Certain fish have reflective tissues that act like biological mirrors, helping them blend into the faint light environment. Silvered sides are common in mid-water animals because a mirror can reflect the surrounding water and reduce contrast. In the deep sea, where light is dim and directional, reflectivity can be camouflage. The physics is delicate. Too much reflection at the wrong angle could reveal the animal. The right reflection can make it vanish. A body becomes optical engineering. Other animals use blackness more complete than ordinary pigment. Some deep-sea fishes have ultra-black skin that absorbs nearly all light, including the glow of bioluminescence. This prevents reflected light from revealing their outline. In a world where even a tiny reflection could betray a predator or prey animal, ultra-black skin is a powerful adaptation. The pigment is arranged in microscopic structures that trap light, not merely color the surface. Darkness becomes not just the absence of light, but a biological technology. The deep sea also contains red bioluminescence in a few specialized predators. Because most deep-sea animals cannot see red, a fish that can produce and detect red light has something like a private flashlight. It can illuminate prey without being visible to them. This is the logic of the dragonfish. Its red light is not dramatic to human imagination because we think of red as bright and obvious, but in the deep sea, red can be secrecy. The same color that warns at the surface becomes concealment in the dark. Bioluminescence can also be used as alarm. Some animals release glowing particles or mucus when attacked, creating a burst of light that may startle a predator or attract larger predators to the attacker. This is sometimes called a burglar alarm strategy. If a small animal is caught by a predator, producing light may summon an even bigger predator that threatens the first one, giving the victim a chance to escape. In the deep sea, light can be bait, camouflage, language, and weapon. It depends on who sees it. Some squid use bioluminescent countershading. Some use ink. Some use rapid color changes. Some use body posture. And some combine all of these. Deep-sea cephalopods can be highly visual and behaviorally complex. Yet many are known from only a handful of observations. A single ROV dive may record a behavior never seen before. A squid holding its arms in a strange posture, a mother carrying eggs, a species changing pattern, a predator using light. Each observation can shift understanding. The deep sea is still in the era where seeing is discovery. There are deep-sea octopuses that look almost ghost-like. Some have fins that resemble ears and are called Dumbo octopuses. They flap these fins gently to move through the water and use their arms and webbing to capture small prey. They live at depths where the water is cold, dark, and under enormous pressure. Unlike many shallow water octopuses, some deep-sea species have different reproductive strategies, producing larger eggs and more developed young. The deep sea changes childhood as well as adulthood. In a place where food is rare, tiny helpless larvae may be a risky strategy. Deep-sea reproduction often favors fewer, larger offspring in some lineages, especially where larvae cannot easily rely on abundant surface food. Larger eggs contain more yolk, giving embryos more resources and allowing young to hatch at a more developed stage. This is not universal, but it appears in many deep-sea groups. The trade-off is clear: produce many small offspring and hope a few survive, or produce fewer larger offspring with a better chance each. In the deep ocean, where chance encounters are rare and growth is slow, evolution often shifts the balance. The life cycles of deep-sea animals are often poorly known because larvae may live in different depths than adults, and adults may be hard to observe. A species found on the seafloor may release larvae that drift in the water column, carried by currents to other habitats. Connectivity between vent fields, seamounts, coral reefs, seeps, and whale falls can depend on larval duration, current patterns, temperature, and chance. This matters for conservation. If one habitat is destroyed, recovery may depend on larvae arriving from another population far away. But if those populations are isolated, recovery may be extremely slow or impossible on human timescales. The deep sea can look timeless, but it is shaped by disturbance. Underwater volcanic eruptions can bury vent communities. Landslides can smother habitats. Oxygen changes can stress animals. Food pulses can create temporary booms. Dead whales can transform a patch of seafloor. Mining tests can leave tracks. Trawling can scrape coral. The difference is that natural and human disturbances operate on different scales and recovery times. Deep-sea organisms evolved with some disturbance, but not necessarily with repeated industrial disturbance across large areas. Bottom trawling is one of the most physically damaging human activities affecting deep-sea habitats. Heavy nets and gear dragged along the seafloor can break corals, sponges, and other structure-forming organisms. In shallow, fast-growing systems, some recovery may occur relatively quickly. In deep coral gardens, where organisms grow slowly and may be centuries old, recovery can take far longer. A single pass of gear can remove habitat complexity that took thousands of years to develop. The deep sea's slowness makes it vulnerable. At the same time, the deep sea is not only a victim in the story. It is an active, living part of Earth. It stores carbon. It recycles nutrients. It supports fisheries indirectly. It shapes climate. It hosts genetic and biochemical diversity. It records geological history. It connects surface life to planetary processes. Calling it unknown should not make it seem empty. Unknown means insufficiently observed, not insignificant. Much of the deep sea is unknown precisely because it is so large, complex, and difficult to study. The deep ocean is also a place of scale. A human body descends through the first few meters quickly. A scuba diver reaches tens of meters. Sunlight fades. Pressure rises. Below 200 meters, ordinary diving becomes impossible without specialized equipment. Below 1,000 meters, sunlight is gone. Below 4,000 meters, most of the seafloor lies in abyssal darkness. Below 6,000 meters, trenches begin. The deepest point is nearly 11 kilometers down. If Mount Everest were placed in the Mariana Trench, its summit would still be more than 2 kilometers below the surface. The vertical scale is difficult to imagine because humans are surface animals. Pressure is also difficult to imagine because we are surrounded by one atmosphere and barely notice it. At 10 meters underwater, pressure doubles. At 100 meters, it is about 11 atmospheres. At 1,000 meters, about 100 atmospheres. At the Challenger Deep, about 1,100 atmospheres. Yet animals live there, not by keeping pressure out, but by being pressure compatible. This is one of the deep sea's quiet lessons. Survival does not always mean resisting the environment. Sometimes it means becoming suited to it so completely that what would crush one form of life simply surrounds another. The cold deep ocean is also a reservoir of dissolved gases. Cold water can hold more oxygen than warm water, but oxygen must still be supplied by circulation from the surface. Deep waters formed in polar regions sink because they are cold and dense, carrying oxygen into the abyss. As that water ages and moves through the ocean interior, organisms and microbes consume oxygen. The deep ocean therefore contains a record of ventilation, when and where water last touched the surface. Measuring oxygen in deep water tells scientists something about circulation, biology, and time. The deep sea changes the meaning of distance. A hydrothermal vent field may be separated from another by hundreds or thousands of kilometers. For humans, that is a long trip. For a larva drifting in deep currents, it may be the difference between finding habitat and dying. For an animal tied to one seep or vent, suitable habitat is patchy, isolated, and temporary over geological time. Deep sea life often has to solve the problem of finding rare places in a vast environment. Vents, seeps, carcasses, wood falls, seamounts, mates, prey. Much of its biology can be understood as a response to rarity. Even the concept of exploration is different underwater. On land, a person can look across a landscape. In the deep sea, the field of view may be only what a light illuminates. Beyond that circle is darkness. An ROV may move meter by meter, recording a tiny path through a huge habitat. A scientist watching the screen sees only a narrow slice of the world. The unexplored deep is not an abstract unknown. It is literally outside the lights. When a new species appears in that circle of light, the moment can be both scientific and strangely intimate. A fish turns its head. A squid changes posture. A crab walks across sediment. A coral polyp opens in the current. The animal is not performing for the camera. It is simply living in a world that had been there long before the vehicle arrived and will continue after the lights leave. The deep sea's beauty often comes from this combination: extraordinary life, seen briefly, then returned to darkness. There is a quiet emotional power in the fact that the largest habitat on Earth is mostly invisible to us. Most of the living space on this planet is not forests, plains, cities, reefs, or mountains. It is dark water. The deep sea is not an exception at the edge of Earth. It is a central part of Earth. Surface life, including human life, occupies the thin, bright boundary above a much larger volume of cold and dark ocean. To remember this is to feel the planet differently, less as a familiar surface, more as a deep body. And yet, the deep sea is not hostile in a moral sense. It is not trying to be terrifying. It is cold because physics makes it cold. It is dark because light is absorbed. It is pressurized because water has weight. Its animals are strange because evolution shaped them for those conditions. The deep sea becomes less frightening when understood, not because it becomes ordinary, but because its strangeness becomes meaningful. The anglerfish, the tube worm, the snailfish, the vampire squid, the glass sponge, the sea cucumber, none of them are monsters. They are successful answers to difficult questions. This is why the deep sea works so well as a subject for a quiet night. It is vast enough to humble the mind, but slow enough not to agitate it. Its mysteries are real, but they do not demand immediate action from the listener. They can be held gently. There is darkness, but also light made by living bodies. There is pressure, but also adaptation. There is scarcity, but also patience. There is death falling from above, but also communities that turn it into life. The deep sea is not empty darkness. It is a slow, living, luminous darkness. Another deep sea mystery is how animals time their lives in a world without sunrise. Near the surface, dawn and dusk provide obvious daily signals. In the deep sea, those signals are weakened or absent, yet many animals still follow rhythms. Some respond to the faint changes in light that penetrate to the twilight zone. Others may follow internal biological clocks, chemical cues, current patterns, food pulses, or the movement of prey above them. Even far below sunlight, the day at the surface can echo downward through behavior and food supply. Darkness does not mean time has disappeared. It means time is carried by subtler signals. Season also reaches the deep ocean. In spring, when surface waters bloom with phytoplankton, more organic material may sink into the depths weeks later. Deep sea animals can respond to these pulses of food with feeding, growth, and reproduction. A bloom at the surface becomes a delayed event at the seafloor, like a message falling through kilometers of water. This delayed connection means that a community living in darkness can still be tied to sunlight indirectly. The deep sea is not independent of the surface. It receives the surface in fragments, particles, bodies, chemicals, and seasons. Some deep-sea animals have skeletons and tissues that are surprisingly fragile when brought to the surface. A fish adapted to high pressure and neutral buoyancy may look soft, watery, or delicate outside its environment. This fragility is often misunderstood as weakness. In reality, it is efficiency. A heavily armored body is expensive to build and carry. In a stable, buoyant, cold environment, gelatinous tissues and reduced bone can be effective. The body is shaped for the water around it, not for air, gravity, and human hands. What seems fragile at the surface may be perfectly suited below. The deep sea also changes the idea of beauty. There are no sunsets there, no green forests, no familiar animal faces, no horizon. Its beauty is in forms of survival. A row of blue photophores, a transparent head, a coral branch growing millimeter by millimeter, a microbial mat feeding a community, a fish balanced on long fins in the current, a shrimp living at the edge of hot and cold water. It is beauty without display for human eyes. Most of it existed unseen for millions of years before any camera arrived. That may be why it feels so powerful when we do see it, because the beauty was not waiting for us. It was simply there. Deep-sea science is still young enough that humility is not optional. Every confident statement has to leave room for the fact that most habitats have been sampled only briefly, some only once, and some not at all. Species ranges may be broader than known. Behaviors may be misunderstood. Abundances may change with season, depth, oxygen, currents, and sampling method. Even basic questions, how many species live there, how connected populations are, how quickly damaged habitats recover, often remain open. This does not make the science weak. It makes the deep sea honest. It reminds us that knowledge can be real and incomplete at the same time. The deep sea asks for a slower kind of attention. A bright animal at the surface announces itself immediately, but a deep-sea animal may reveal its meaning only when you understand the pressure, the chemistry, the darkness, the scarcity, and the history around it. The odd mouth, the pale body, the strange light, the long lifespan, the patient posture, each feature is part of a larger answer. The more carefully you look, the less random the strangeness becomes. It becomes adaptation, and adaptation becomes story. There is something about the deep sea that speaks directly to the part of the mind most awake in the hours just before sleep, the part that can hold large and uncertain things without needing to resolve them, that can rest in the presence of what is not known without demanding answers. The deep sea is the largest dwelling place of life on Earth, and it remains mostly unvisited, partly unmapped, and far beyond ordinary human experience. The creatures that live there, the anglerfish with its dangling bioluminescent lure, the vampire squid in its oxygen-poor twilight, the tube worm harboring chemosynthetic partners in its body, the snailfish pressing against the limits of what a vertebrate can endure, are not legends. They are alive right now, in the dark, in the cold, under pressures that would collapse air-filled spaces and make unprotected human survival impossible. The deep sea will be there in the morning. It will be there next year and next century. It holds its mysteries with a patience that makes human urgency seem brief and gentle. And from here, from the surface, from the warmth of wherever you are resting tonight, you can let that dark and quiet immensity be simply what it is, real, enormous, still partly unknown, and going about its extraordinary business entirely without your attention. Somewhere in that darkness, a lanternfish is rising with the night. Somewhere, a brittle star is crossing soft sediment. Somewhere, bacteria are turning chemical energy into food beside a vent. Somewhere, a whalebone is feeding worms and microbes. Somewhere, a coral older than a city is filtering the current. Somewhere, A snailfish is moving slowly through trench water under a pressure almost impossible to imagine. None of it requires you to solve it tonight. You can simply know that it exists. You can let the thought of it become wide and quiet. Sleep well. Good night.